Good morning, friends. Today the topic of discussion is biomolecules. As you all know that biomolecules is a very basic area of biochemistry, and often I have seen that students get very much confused regarding the chemistry of biomolecules. So in today's tutorial class we will discuss about the different types of biomolecules, and this topic will be covered in a series of lectures. So this lecture deals with the introduction of biomolecules. So let's first ask that what are biomolecules? As the name implies, there is a term bio. Bio means living. So these molecules are present in any cell, and the cell has to be a living component. So within the cell, if we see, there are various types of molecules. So these molecules may be of different types, and when we want to visualize the chemistry of a cell, the component biomolecules are very important. So when we look into the cell, the total number of biomolecules which are present within the living cell is called as the cellular pool. Okay, so whenever you get a term describing cellular pool, it means that it comprises of various types of biomolecules. And each and every biomolecules always have a definite function. So their main action is to play different important physiological roles. Now broadly, biomolecules can be of two types. They may be of two types: organic and inorganic. In organic biomolecules, we find like proteins, the nucleic acids, and as you know, nucleic acid means the DNA component and the RNA component. Then there are the polysaccharides. Polysaccharides are a type of carbohydrates. They are basically complex carbohydrates. And there are lipids. Lipids which are commonly termed as fats. In case of inorganic biomolecules, there are broadly different minerals which exist in the form of ions. Okay, so there are definite minerals for plants and definite minerals for animals. And the number of mineral ions is very important for the growth and development of both plants and animals. Now this is the classification of biomolecules depending upon the organic nature or the chemical nature, that is organic and inorganic. So now the next question is, how are you going to find out or know that what kind of biomolecules are present within a cell? So if generally this area is broadly dealt in competitive examinations, like they ask you a question that you are given a cell and you are given some chemical reagent without using any instrumental procedure, how are you going to find out the different types of biomolecules within the cell? So the method is very easy. So this is the detection part. Okay, how are you going to detect out the different types of biomolecules within a living cell? So for this, what you do, you take a living cell. Okay, to this living cell you add trichloroacetic acid. Which is commonly known as TCA. Okay, so you take the living cell, you take trichloroacetic acid, and you use a mortar and a pestle to grind them. You grind the cell. So after you grind the cell, what do you get? You get a slurry. So you take the slurry and you filter it out. How do you filter it? You do one thing, you take a funnel and you put the slurry within the funnel, okay, and you filter it. So there is a portion which you get here. This is called as the filtrate. Okay, this filtrate comes downwards and there are some materials which remain here. Okay, so this filtrate is called as the acid soluble part. Acid soluble part, or you may call it as the acid soluble fraction, or you may call it as acid soluble part. Anything, it's your wish. And in this portion you get the acid insoluble part. In the acid soluble part, you broadly get the biomolecules having a molecular weight within the range of 18 to 800 Dalton. However, in some books you may find it as 1000 Dalton. Okay? So, this fraction has got solubilized in trichloroacetic acid, and this portion contains macromolecules having the molecular weight in the range of 18 to 800 Daltons. Okay? And this portion, which is the acid insoluble part, this portion has the molecules which have the molecular weight of more than 10,000 Dalton. So this is a method of separation of biomolecules on the basis of molecular weight. You see, this is the fraction which has the molecules having a lower molecular weight, and this is the fraction, the acid insoluble part, has molecules having higher molecular weight. Okay, so this portion is called the microbiomolecules. Okay, these are called as the microbiomolecules, and this is known as the macrobiomolecules. Okay, so here previously I had given the basic classification of biomolecules as organic and inorganic. So now this classification is based on molecular weight. This is the macrobiomolecules, and this fraction which has the molecules of 18 to 800 Dalton are microbiomolecules. Now in the macrobiomolecules, you get the proteins, you get the nucleic acids, that is the DNA and the RNA, you get the polysaccharides. Okay, polysaccharides are a group of complex carbohydrates. Okay, so these are the macrobiomolecules. In case of the microbiomolecules, what do you get? You get the amino acids. Okay, you get the nucleotides. You might also get the peptones. Then you might get the rest carbohydrates like disaccharides and oligosaccharides. Okay. So you see that in the case of carbohydrates, polysaccharides are only macromolecules. And in microbiomolecules, the disaccharides and oligosaccharides are the carbohydrates which are microbiomolecules. We have left out one particular biomolecule, that is lipid. In case of lipid, you get it here. You should get it here. Okay? But you see that in case of the lipid, the molecular weight is about 1000 Dalton. Okay, so lipid has a molecular weight of about 1000 Dalton. So you should get lipids within the microbiomolecules, which you do not get. So instead of getting lipids here, you get the lipids here. Okay, I am writing it with a star. You get the lipids here. So why is the case so? If you follow the definition of the method of separation here, it is based on molecular weight. Okay, so you should have got your lipids here because the molecular weight is 1000 Dalton, but you get it here in the macromolecular fraction. It is because you know that lipids are often attached onto the plasma membrane or any membranes. Lipids are normally not freely found. So when you grind the cell, the portion of the membrane having the lipid turns into smaller vesicles. Okay, and these vesicles are insoluble in aqueous medium. Okay, so we are grinding it with what? Trichloroacetic acid, and trichloroacetic acid is a dilute acid, and dilute acid means that there is water in it. And lipid, by its nature, because of its amphibactic nature, it forms a small vesicle, and that vesicle does not solubilize in the aqueous medium. So those vesicles, due to their insolubility, are not found in this filtrate portion which has the microbiomolecules. Instead, they are found in the macromolecular portion. So it should be clear now that although lipids are categorized under macrobiomolecules, biochemically, depending upon the molecular weight, they are actually microbiomolecules. Okay, this is due to their insolubility. So this gave an idea of what biomolecules are. Broadly, there are two types depending upon their chemical nature, that is organic and inorganic, and depending upon their molecular weight, that is microbiomolecules and the macrobiomolecules. So in the series of lectures, we will discuss one by one the different biomolecules. My next video will discuss about the different types of